So, good afternoon, students.
Good afternoon, sir.
So, any question, any query from the previous class? So, anyone, any question, query? No issues? Okay, so let me just show you a video of what we have learned in the last few classes. So I hope you remember we started with the historical aspect of vaccines, right? Because when we talk about immunology, it starts with the vaccines, right? So let me show you a video. It talks about um, the history of immunology uh, from the smallpox eradication till this time when we have seen this COVID. So let's first see this video, and after that we will start with the history of immunology. So right now you have seen how vaccination process developed, right? But then later on, core immunology developed, like the development, analysis of antibody, the humoral immune response, cellular immune response, transplant immunology. So after this video, we'll talk about all those things. So let me share the video.
Sir, it's not audible.
Should be audible, I don't know. Let me share again, okay?
Okay.
The idea is to limit the spread of infectious disease throughout the population, and
Yes, sir, it's audible.
don't have the option of getting vaccinated. However, people cling to the notion that vaccines are harmful, and anti-vax sentiment persists. One of the most severe viral diseases known to humankind is smallpox. Even into the 18th century, 500,000 Europeans died of smallpox every year. And even into the 20th century, it was still claiming 300 to 500 million lives. Some people who caught the disease survived. And those people were now immune to the disease. By the Middle Ages, the Chinese had developed a technique to manufacture immunity. They collected scabs from an individual with a mild case of smallpox, allowed them to dry, ground them up into a powder, wrapped the powder in cotton, packed the material into a pipe, and puffed the whole concoction into an uninfected person's nose. This process is called insufflation. This is the first documented use of inoculation, which is a deliberate infection for the purpose of developing immunity. In the 18th century, this practice was introduced to Europe, and the practice becomes known as variolation in England in honor of the scientific name for smallpox, variola. Edward Jenner was a physician in England in the 18th century. Jenner knew about the practice of variolation. He himself actually had been variolated when he was 8 years old. He also knew something else, which was pretty widely known in Europe at the time, but was relegated to the status of folklore or an old wives' tale. And this something was that cows contracted a disease similar to smallpox. This disease was called cowpox, but it was much milder. Cows didn't die from it. And it was also common knowledge that milkmaids who worked with cows sometimes acquired cowpox, and these milkmaids were then immune to human smallpox. In 1798, Jenner seizes the opportunity to inoculate a handful of friends and neighbors with cowpox. He again variolates them with smallpox subsequently, and he finds that not a single one of them shows any sign of having been exposed to smallpox. He coins a scientific term for cowpox. He calls it variola vaccina for the Latin word vacca, meaning cow. And his procedure he calls vaccination. People keep vaccinating, and in 1980, the World Health Organization declares smallpox eradicated. There are religious objections. There is resistance to deliberately infecting healthy people. But with vaccination, there's now the added consideration that we're using animal material. Some consider it unclean, others consider it inhumane. There's also risks that the procedure is transmitting other diseases. This was actually a big risk at the time. Another aspect of the controversy that began even before Jenner's time with the early practice of variolation and still continues today is the tug of war between mandatory vaccination and personal liberties. The problem is that some people can't be vaccinated because their immune system is not healthy enough. These immunocompromised people include people with disorders of the immune system, or very old people, or newborn infants rely on herd immunity to protect them from disease. In modern times, our generation doesn't remember how devastating the effects of these infectious diseases were. We never experienced smallpox. We never even experienced measles, mumps, or rubella. The fear of the harmful effects of vaccines sometimes feels more immediate than the harmful effects of the diseases themselves. Smallpox was a scourge on humanity for millennia, but there was a glimmer of hope. Although millions of people succumbed to the virus, some people who caught the virus survived, and those people were now immune to the disease. We are still advocating strongly for vaccination as a public health measure.
Okay, so students, any questions from this? Okay, so let me continue further. Okay, let me share the slides. Okay, so I hope the slides are visible to you, right? So we have already seen the initial part of the history of immunology, right? It all started with the formation of vaccine, right? So again, there are few slides on that, but let's understand immune. Basically it means, so it's a Latin word which means immunus, okay, which actually means to be free or exempt, right? So immunology, basically the origin is from this Latin word immunus, right? And you all know by this time that immunology, it's the study of the physiological mechanisms that humans and other animals use to defend their bodies from invading organisms. Okay, so the term immunity was first used in 1775 by Van Schwitten. He was a Dutch physician, okay, and he termed this as immunitas to describe the effects induced by an early attempt at variolization. So now you also understand what is variolization, right? So, and immunity, the term, it's basically the state of our body to protect from infectious diseases with the help of the immune system. Okay. So I hope by these two terms you understand what is immunology and what is immunity, right? So immunology is basically the study of the immune system, and immunity is the body's state, right? Like I'm immune, or I'm having immunity against a particular organism. So immunity is basically the state of our body, and immunology is the study of the immune system. Okay. Now, when we talk about immune system, it's basically the third line of defense against infection, right? Immune system uh, plays a role at the third step, right? So if we see the different steps, so there are non-specific defense mechanism, and then there is specific defense mechanisms, and this basically include our immune system, right? And I will make you understand these specific non-specific things. Now let's first see the first line of defense, right? Now when we talk about the first line of defense, the most important and the biggest organ is skin, okay? And why skin is non-specific, why it is placed here in non-specific? Because see, the skin will not see whether it is a bacteria, whether it is fungus, whether it is protozoa, whether it is helminth. It will attack on all, right? Or skin will prevent the entry of all these organisms. So therefore, it is non-specific. Okay. Similarly, you have mucous membranes. Okay, so you you know there are a lot of mucous membranes in our nose, mouth, right? So, and then the secretion of skins and the secretions in these mucous membranes. So these secretions which are there in the skin and mucous membranes, they are antimicrobial in nature, okay? They also have some components of the immune system in them, okay? They have a lot of degradative enzymes. So they basically act on whatever is coming in contact with it, right? So you have a skin, it will protect you. It is the first line of defense, right? So if a pathogen has to cause disease, it has to enter through your skin, right? So nothing can enter until there is some problem with the skin. So suppose you get a cut, you get a burn, right? So when the skin is not there, in that case, the pathogens will enter into your body, right? Then obviously you have nose, the pathogens can enter through the nose. So for their that kind of protection you have the mucous membrane. So if a pathogen enters into our nose, okay, so the mucus secretions, they will trap the pathogen. Okay, they are sticky in nature, they will trap the pathogen, and in those secretions there are antimicrobial agents, there are a lot of enzymes, they will all attack and kill the pathogen that has entered into our body. Okay, so they are all the first line of defense, right? Now let's see the second line of defense. So this include the white blood cells. Okay, these are the phagocytic white blood cells. So I hope you understand what are phagocytic. So phagocytic basically means, suppose this is a bacteria, it has entered. So let me take this example. So I have this key. This is the pathogen, right? Now it has entered into our system, right? Now what the phagocytic cells will do, they will sense the presence of a foreign body, they will move towards this foreign body and they will phagocytize this pathogen, right? So these are the phagocytic white blood cells. Again, they are non-specific. So they will phagocytize whatever comes in their way. It may be bacteria, it may be virus, it may be fungal spore, it may be some protozoal spore, it could be anything. Okay, so they will just go and phagocytize it, and therefore they are non-specific, right? Then you have antimicrobial proteins. As I said, in our system, there are a lot of antimicrobial proteins. They will again uh, kill the pathogen, okay, because they are antimicrobial, against the microbes. So these proteins, these enzymes, they will go and bind to the pathogen and they will kill the pathogen, right? And then the inflammatory response. Again, you all have seen inflammation, right? Whenever you get a cut, there is redness, there is pain. So all those symptoms of inflammation. And again, it is non-specific whether a fungus has entered, whether some bacteria has entered, there will be inflammation, right? So this is the first line of defense, which includes skin, mucous membranes, secretions of the skin and mucous membranes, and there are various components in the skin, various components in the mucous membrane, right? So we may talk about that later, right? And then second line we have already discussed. Then comes the third line, which is very specific, and it includes the antibodies, right? And I hope by now you understand what antibodies are, how they look like, what they do, right? But yes, we will be talking about antibodies in much more detail later on. And then you have the lymphocytes, like your T cells, right? And there are different types of T cells. So you have the T helper cells, cytotoxic T cells, T regulatory cells. So a lot of different type of T cells. Okay. So uh, when we see immune response, so one of the thing is recognition. Okay, immune response or the immunity should be able to recognize between self and non-self. Okay, so the immune system should be able to recognize that this is self protein and I don't need to attack it. Okay. And it should be able to recognize, okay, this is a pathogen which has come into my body from outside and this need to be eliminated, right? So one of the most important factor of immune response is the recognition of self and non self. Okay, क्योंकि अगर ये recognition नहीं होगी, then the immune system will start attacking the self cells, okay, which can create problem. And one of the condition is the autoimmune disorders. We will talk about that later. Okay, another is effector response. Okay, it's just not about recognition. Okay, the immune system recognize that something has come from outside. Okay, that is recognition. बट सिर्फ रिकॉग्नाइज करके नहीं छोड़ना है, राइट? there has to be some effector response which can lead to elimination of the pathogen, right? So pehle you will recognize, not you, basically the immune system will recognize that something has come from outside, it's non-self, okay? Then an effector response has to be generated so that it leads to elimination of the pathogen. And then the third most important factor is the memory response, right? आपने recognize कर लिया, आपने एक बार उसको eliminate कर दिया, and then you also need to remember, okay, if this pathogen comes again, how will I fight, right? What are my uh, tools or weapons to kill that pathogen, okay? And as I said earlier also, in case of vaccines also, uh, when we encounter the pathogen for the second time, okay, in that case, the immune response is rapid, right? And it is heightened, okay? So it is more robust immune response and more rapid immune response, right? Because obviously when this immune system is fighting a pathogen for the first time, okay, it has to recognize, it has to like create a network, right? It has to give signals, and then the whole process will start, right? But if there is memory, right, so immediately when the immune system sees the pathogen, it comes rapidly and respond and eliminate the pathogen, okay? So this is all the three major things that the immune response should do. One is recognition, effector response. Effector response basically means effective response in elimination of pathogen, and then a memory response should also be generated, okay? So we consider immune system as friend, but in some cases it may act as foe, right? It's, for example, autoimmunity. When the immune system, it starts to attack the self cells, okay? Another example is that of hypersensitivity, right? Like the allergies, okay? In that case, the immune system or the inappropriate immune response lead to these hypersensitivity reactions, right? And sometimes these reactions, the anaphylactic reactions, can be dangerous, okay? They can lead to death. So mostly immune response, immune system is a friend, but in some cases it may act against us, okay? So we will understand both autoimmunity as well as hypersensitivity in our classes. So obviously that will come at a later end of the semester, because before that we need to understand the whole components of the immune system, okay? Only then you will be able to understand autoimmunity and hypersensitivity. And students, I am telling you at this moment, to understand autoimmunity, hypersensitivity, and other advanced things, you will have to understand the basics, okay? So suppose I am talking about IgE, right? And by that time, if you have forgotten how IgE look like, then it would be difficult for you to understand those concepts, okay? So you have to keep reading immunology, right? From the initial stage itself, okay? So that by the time you reach to the advanced modules, you don't find it difficult, okay? Now, if we talk about this, so the disease plague, it was observed like 430 BC, so it has been observed even in the mummies, right? Um, so I'm not going again into this variolation thing because you have already seen this, okay? So I'm not going to again get into this. Uh, I'm sure you know Lady Mary Wortley Montague, right? She was a famous poet at that time. Herself, she was a smallpox victim, okay, and she has written a poem also, The Smallpox. And in 1715, smallpox ruined her good looks and left her without eyelashes and with deeply pitted skin. So you have seen the pictures how. So when those vesicles they are healed, right? Then there is a small pit kind of uh, surface on the skin, right? So she was wife of the British ambassador to Turkey, and she learned how to variolate people. So she was there in Turkey. She saw the process of variolation, and she variolated herself, her son, as well as a daughter, and she was the one who told this in England, okay, and the permission to vaccinate the children of the Prince and Princess of Wales, okay. So that experiment was called the Royal Experiment, okay, and because of that, this method of variolation was adapted in England and other parts of the Europe. So because she was the ambassador, a wife of ambassador, right? So she saw that in Turkey, and then she went back and told to the Europeans that this is a process by which you can protect your children with smallpox. And later on, this story of Edward Jenner we all know. Uh, So, after years of experiment, Edward Jenner carried out a famous experiment on healthy 8-year-old boy named James Phipps. Okay, so this I have already shown you in the pictures. Okay, so I'm skipping all this. You have seen this picture. So Edward Jenner inoculating James Phipps with cowpox, and that gave him protection against smallpox. And you can understand the importance of this vaccine. So during the Franco-Persian War, okay, so there was smallpox epidemic. French army was not vaccinated, and 23,400 soldiers died, whereas the German army was vaccinated and only 278 died because of smallpox. So you can see French army lost half of the battle because of smallpox, right? So almost 24,000 soldiers dying because of smallpox. It was a big number. So with this, you can understand the importance of vaccine, and people also started to realize the importance of vaccine, right? So again, we have discussed the last reported case of smallpox. It was Ali Macken in Somalia. Okay, there were few more cases, but they were like accidental lab leakages. Okay, and in 1980, ultimately WHO declared the world and its people free from smallpox. Okay, so you can see the numbers went down after vaccination, and ultimately in 1978 or 1980 the world was declared free of smallpox. Okay, and as I said, last stocks of the smallpox virus, they are kept at Center for Disease Control and Prevention, Atlanta, USA, and in Russian State Research Center for Virology and Biotechnology in Russia. Okay, now we have seen vaccines. Now let's see some other things. So Robert Koch, okay, and Louis Pasteur, they established bacteria as cause of disease. So they were the two scientists. They did a lot of work, right? And they established that bacteria is responsible for causing diseases, right? So what Pasteur did, he injected animals with live attenuated microorganisms, okay, so cholera, anthrax, rabies, okay, So that was another thing. So he uh, did that attenuated work, right? What we call as attenuated vaccines. Okay. So Robert Koch, he was a German physician, started to work on anthrax, right? And identified for first time the causative agent of an infectious disease. So it was the work of Robert Koch. So he was working on the anthrax disease, and he identified for the first time that the causative agent is a bacteria. Another important thing is the Koch's postulate. Okay, so he postulated that some conditions are there that must be satisfied before accepting that particular bacteria cause a particular disease. I don't know if you have read Koch's postulate. I think you have read this in microbiology, right? So you understand that he said that there must be some conditions. Okay, if those conditions are fulfilled, then it means that that particular organism is responsible for causing that disease. Okay, but yes, in some few examples, this postulate may not work, right? Only in few very rare cases. And Robert Koch, he also discovered the tuberculosis bacilli, and he was awarded a Nobel Prize in 1905. Then another scientist is Louis Pasteur, and he worked on developing of the immunity to cholera, right? So there is a story of his work. So what happened in his lab? Now this is again a very interesting story. Um, he was working on this chicken, okay, and in his lab there was aged bacterial culture, okay. So he just kept it, and over a period of time, so he just kept that, okay, and then he had to inject that culture in the chicken, okay. So what he did, he injected this aged bacterial culture in chicken, and then he saw that there was no disease and no chicken died even when this cholera pathogen was given. Okay. Now he obviously he thought there was some problem here with this culture, so he again took fresh bacterial culture, okay, and then he injected in two different groups, one in these no disease wala group, okay, and another one जिनको ये वाला culture नहीं लगा था, okay. So then he observed that the chicken having which were already inoculated with this aged bacterial culture, they survived, right? Whereas the ones which did not receive this aged bacterial culture, they died. Okay, and the whole process was later on understood, and it was the attenuation. Okay, so because of this aged bacterial culture, or because the bacteria with time lost its virulence, it became attenuated, okay, which means it was live but it was not able to cause any disease, okay. So this was the contribution of Louis Pasteur. So from here it came into existence that you can attenuate an organism, okay, and that can act as a vaccine, right? Okay. So, Pasteur's contribution, so changes in hospital practices to minimize spread of disease, because now people understood that it is the bacteria which is causing diseases. So there were some changes in hospital practices. He discovered that weakened form of microbes could be used as immunizing agents. Okay, जिसको हम आज की डेट में attenuated vaccines कहते हैं. So he discovered this aspect that a weakened microbe can be used to immunize a person. Okay, he found that that rabies were transmitted by agents so small that they could not be seen under microscope, right? So by that time the scientists were able to visualize bacteria, and he was also working on rabies, and he realized that there is even something smaller than bacteria which is causing disease, right? So thus revealing the world of viruses. Okay, so he actually did not show any virus, but he said that there is something even smaller than uh, bacteria which is causing this disease, rabies. Okay, and later on we know that it is a virus that is causing rabies, right? He developed the process of pasteurization. We all know what is pasteurization. It's a process by which harmful microbes in perishable food products are destroyed by heat. Okay, so we know this process, pasteurization, without destroying the food. So he developed this practice of pasteurization. So now that increased the shelf life of the food products, right? Just for example, milk, if we pasteurize it, we boil it, right? So the shelf life increases, right? And as I said, he also developed attenuated vaccines for cholera, anthrax, and rabies. Now, let's see the Pasteur's anthrax experiment. He first vaccinated one group of sheep with heat attenuated anthrax bacilli, right? ध्यान से सुनना. One group of sheep with heat attenuated bacilli, right? He injected virulent cultures of bacilli in vaccinated and unvaccinated sheep, and all the vaccinated sheep lived and unvaccinated died. So I think you can understand this. So he took the sheeps, okay, and injected attenuated ones, okay. Later on, he took this sheep and took the unvaccinated sheeps, okay, and again injected okay. So he injected uh, the pathogen in both these groups, and he observed that all sheeps which were injected with attenuated bacilli, they survived, whereas the unvaccinated sheeps, they died. Okay, so this developed the concept of attenuation, and that attenuation can be used for uh, generating an immune response. Okay, okay students, I'll just stop here for your queries. So if you have any queries, you can ask or you can discuss if anything you want to discuss. Okay, students, you can also see the assignment and start doing your assignment. So any question?
Excuse me, sir.
Yes.
Sir, actually, at my end, it's showing that your assignment is already being submitted.
How come?
Um,
Is everyone seeing the same thing?
No, sir.
No,
sir,
sir.
I will check at my end.
Okay. Because as such, I have not received any response yet. So nothing is submitted to me yet. Okay. Okay, students. So by the time you are doing your giving your exam, that test, let's have your attendance as well. So be ready for your attendance. Adish,
Present, sir.
okay, Adish, Ayushi,
Sir,
okay, Atoeba,
Present, sir.
Sam,
Present, sir.
Devanshi,
Present, sir.
Ishita,
Present, sir.
Janvi,
Present, sir.
Kshitij, okay, Manaswani,
Present, sir.
Namrita,
Present, sir. Present, sir.
Nandini,
Present, sir.
okay, Nehal,
Present, sir.
Rupal,
Present, sir.
Sahiba.
Present, sir.
Again, you have some network issue today.
Yes, sir. Because of the weather, I'm having some problem with my Wi-Fi.
Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani, students also keep submitting your responses. Only 4 students have submitted their response yet. Okay, so keep submitting your responses. Tanvi,
Present, sir.
Tridev,
Present, sir.
Vivek, okay, Yukta,
Present, sir.
okay, and Yuvraj.
Present, sir.
Okay, okay, fine. Okay, so everyone is present. So let's see how many of you have submitted. Okay, only half the class has submitted the responses. Students, do it fast. So 14 students have submitted. Rest of you do it fast. Okay, let me discuss the answers. I think today it was really easy, right? So the first question was the term immunity was first used by, the answer is Van Switten. Okay. Question 2 was attenuated vaccines for cholera, anthrax, and rabies were developed by, the correct answer is Pasteur. Okay. Question 3 was Eli Metnikov identified. So he identified macrophages. Okay, correct answer is macrophages. And question 4 was last reported case of smallpox was. The correct answer is Ali Makin. Okay, but a lot of you have attempted this wrong. Many of you have written none of the above. Few have mentioned Ali Bocaro, but the correct answer is Ali Makin. Okay, Ali Makin was the last case of smallpox. He was from Somalia. Okay. So with this, I'll end the class now, and if you have any queries, questions related to today's class, we can have it in the morning itself, because I'll be having the first class with you tomorrow. Okay. So see you students tomorrow morning.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you.